The title of the talk is Basic Genetic Principles for the Non-Geneticist. And just imagine genetics having started as an observational science. So Gregor Mendel looked at different peas and different colors and shapes, and then drew some conclusions regarding inheritance pattern. And similarly, human genetics began as an observation, looking at different traits and how they were inherited. In this particular picture, you can see how bad taste in clothes is inherited as a pattern. But with the discovery of DNA, we are beginning to understand better how inheritance actually works from the chromosomal, from the molecular point of view, and then genetic becomes an interventional discipline. To understand better how aberrations in the genetics cause different human conditions, it's best to envision the human genome as an encyclopedia. Now, having made the slide, and I recognize that some young people have actually don't really know what an encyclopedia looks like. So it's just like Wikipedia, but divided into books. But for those of you who are old enough to know what an encyclopedia is, just imagine that we inherit from our parents a set of 23 pairs of chromosomes. There are just like 23 volumes, pairs of volumes, where each chromosome is one volume, and that each chapter is one page. It's actually a recipe for making a product that the body needs for proper development. And just like a recipe is written in letters, so is a gene written in certain letters. They are A, C, T, and G. And the entire genome has about 3 billion base pairs, 3 billion letters. It's just what a regular thumb disk or a disk on key contains, so 3 giga. But only about 1 to 2% of these actually encode for the genes themselves. The rest is, we sometimes don't know what it is. And then this is encoded, it's translated by the genetic code. So a basic gene has certain properties. It has exons, which actually encode for the protein which is produced usually, and intervening introns in between. And the gene is written in DNA code. It is translated into RNA or transcribed into RNA. The RNA is edited for a messenger RNA, and the messenger RNA then is employed by the ribosomes to produce the protein that is needed for the body. And one can think of the protein as the building blocks of the body. Some of the structural proteins like collagen and elastin, hormones, many hormones are proteins, enzymes, receptors, and the other components of the bodies like fat and carbohydrates are metabolized by enzymes. So basically proteins make up the entire component of the body, or else produce it. So having genetic aberrations may occur at different levels in the hierarchy structure of the genome. So we can have aberrations at the chromosomal level, we can have aberrations at the submicroscopic level, and then we can have variations or aberrations in the actual sequence themselves, and I will walk you through them just to get an idea of what we're looking at. So first, looking at chromosomal aberration, this is just like having an extra volume or a missing volume in our encyclopedia. And this may be ascertained by cytogenetic analysis. This will be described more in detail by Vincenzo Ciriano in the next talk, and using technologies that is virtually unchanged over the last decades. We can look at with the microscope, see the chromosome, count them, and so on. That allows us to check for numeric aberrations, different aneuploidies like trisomy 21, Down syndrome, trisomy 18, Edwards syndrome, trisomy 13, different sex chromosome abnormalities, all have to do with an added chromosome, and monosomy, having a chromosome missing, like monosomy X or Turner syndrome. These are all called aneuploidies. We can also detect mosaic aneuploidy. A mosaic aneuploidy is a situation where within the body there are at least two cell lines that have different chromosomal structures. For instance, Turner mosaic will have 46 chromosomes, and then in some cells, but only 45, and a missing X chromosome in others. Or trisomy 21 mosaic will have 47 chromosome with an extra chromosome 21, but only in some of the cells. And the consequence of such mosaics depends on what chromosome is involved. Some chromosomes are more harmful than others. The percentage of abnormal cells, their distribution through the different tissues, and other ramifications. So we'll hear more about mosaicism in Josh Blazek's talk having to do with PGS, and then tomorrow in Francesca Sgrati's talk, seeing the implication of mosaicism in prenatal diagnosis. But one must remember, and this is a quote from Gardner, which is the kind of the Bible for cytogeneticists, that the majority of mosaicism identified at prenatal diagnosis, and indeed probably in preimplantation genetic screening, does not presage an abnormal baby. So this must be taken into consideration. Mosaicism may occur in varying due to different mechanism. Most mosaic forms have to do with mitotic non-disjunction. That means that the embryo at meiosis begins. Do we have a pointer?
On the left.
Yeah. I hope you can see with the green. Mitotic, the gametes are normal, male and female. The zygote is normal. But then during cell division subsequently, some of the cells have a misdivision of the chromosome, whereas one cell gets an extra chromosome. The other one with the missing chromosome usually is annihilated. It does not propagate. So in this situation, you have some cells that are normal, but some cells that are abnormal. This is called mitotic non-disjunction. And we also have meiotic non-disjunction. This is normal male gamete, but an abnormal female gamete. So the zygote that develops has 3 chromosomes, an extra chromosome. That will create a trisomy, but sometimes some of the cells lose the extra chromosome. So in this case, you still have a mosaicism. Some of the cells retain the trisomic chromosome, the extra chromosome, but some of the cells self-correct. So that is another mechanism for mosaicism. We'll hear more about this in the next talks. Of course, chromosomal aberration may also be structural in origin, like translocation. In this situation, some of the, say, 2 chromosomes exchange a bit. So you have a balanced translocation here. This is like taking some pages from one book and moving them to another book and taking some from the other book and putting it in the next one. This is usually harmless if it's balanced, but sometimes in the offspring of such individuals, we have unbalanced translocation, which may lead to miscarriage, an abnormal offspring, and infertility. And we'll hear more about that in some of the PGD talks. We also have diving deeper down into the genome, we have some submicroscopic changes. These are changes that cannot be detected using the standard microscope, requiring additional technologies like FISH or microarrays, and we'll hear more about that in the next talks. This is just like having a book with a few pages torn out. So just by looking at the book, we can't say that there are missing pages. We have to count one by one. So this requires more elaborate technologies, which we'll hear about. And initially, there were some well-described phenotypes like DiGeorge syndrome, Williams syndrome, Prader-Willi. But then now we recognize that there are hundreds and hundreds of different conditions, some without an actual name, and more are being discovered with the use of microarrays. And we'll hear talk about this from Ron Wapner tomorrow. And this is an example of microarray technology. and some of the abnormalities that may be detected, like DeGeorge syndrome or 22q deletion using a microarray technology. Then lastly, if we go down as deep as the single letter in the sequence, we may find certain changes, certain sequence variants. And this is like having in the book some word misspelled. One letter is exchanged with another letter, or a few letters are missing. And that sometimes may cause an aberration, sometimes not. And these will be called sequence variants. In the past, we used to refer to changes as a mutation. But according to the recent American College of Medical Genetic Standards and Guidelines, we're not to refer to each change as a mutation, rather is a graded system where something that causes real damage will be called a pathogenic variant. Something which is likely to cause will be called likely pathogenic variant with greater than 90%. Some variants we're not really sure what they mean. They're variants of unclear significance. And then some variants are likely benign or completely benign. So just to give you an example, say we have a sequence here and the letter A is exchanged with the letter T. What is the implication? That has to be seen in a certain context of which it's written. So just like using the word gray with A versus the gray with E, they're both acceptable. This change doesn't really change the meaning of the sentence. So this would probably be a benign variant. Of course, we can have a likely pathogenic variant. Here, again, one letter is exchanged, but then instead of using one big egg, if we use one bug egg, that certainly we don't get the product that we wished for. So that is a likely pathogenic variant. It has to be seen within the context. And we'll hear more about ways of detecting variants using next generation sequencing with Mark Hughes' talk and certain other talks tomorrow. So the implication of a pathogenic variant, just imagine you have the wild type gene here in green, and the product of which will be the normal protein. If you have a pathogenic variant which changes the amino acid composition, such as a mutation, that will cause the protein to bend. So let's see, as an example, using the CFTR protein, which is responsible for cystic fibrosis. The normal protein encodes for a channel which allows chloride ions to move from within the cells to the outside of the cells of the respiratory and digestive system. With the chloride ions, water comes out, and that dilutes the secretions of these tracts. However, if we have a mutation, the protein is misfolded. So this blocks the channel, not allowing the chloride ions to leave the cells. Therefore, the fluid, the water, does not follow them. And the secretions of the tract, of the respiratory tract or the digestive tract, the secretions become very viscous, they become very viscous, and that causes the condition called cystic fibrosis. Next, I'll give a short introduction about different modes of inheritance. First one, autosomal dominant. With an autosomal dominant condition, if you have an affected parent, and that parent needs only one abnormal copy of the gene to have the condition, and if the partner is healthy and has two normal copies, the offspring will, half of them will be abnormal, having inherited one abnormal copy of the gene, and half of the offspring will be normal. And this is a typical pedigree. We see a transmission from generation to generation. Both sexes are affected, and then half of the offspring of each individual have a likelihood to have inherited the gene. And some examples like neurofibromatosis or tuberous sclerosis, the common achondroplasia, Marfan syndrome, and many cancer predisposition genes like familial adenomatous polyposis or BRCA1 mutations and so on. Next one is autosomal recessive. With autosomal recessive inheritance, one needs to have two abnormal copies of the gene to be affected. And usually, an affected offspring, having inherited two abnormal copies, has two healthy parents. The healthy parents each has one abnormal copy in dark blue and one normal copy. The normal copy is enough for them to be healthy. In this situation, such parents who are carrier parents will have 25% chance of having an affected child, 50% having a healthy carrier like themselves, and 25% having an unaffected non-carrier. And many of these conditions, we will find that the parents of affected children are consanguineous. And screening for such autosomal recessive condition is something that is becoming more and more common, and we'll hear about expanded carrier screening tomorrow from Jim Goldberg and from Julio Martin in their talk in the early sessions tomorrow. And some examples of such autosomal recessive condition like cystic fibrosis and spinal muscular atrophy, Tay-Sachs, thalassemia, sickle cell anemia, and most of these conditions you will have heard of already. X-linked conditions are inherited strictly from the mother. The mother has two X chromosomes. The father has one X and one Y chromosome. An unaffected father will give the X chromosome to all the girls and the Y chromosome to all the males. And the mother who has an X with a mutation in it will have half of her daughters will be carriers and half of her sons will be affected. And this is a typical X-linked recessive pedigree with affected males only, and they're connected to each other through maternal lineage. And some examples include Duchenne and Becker muscular dystrophy, the hemophilias, color blindness, G6PD deficiencies, and many others. And then lastly, X-linked dominant conditions. These also inherited from the mother, but affecting both male and females because having an abnormal copy of the gene on the X chromosome is enough to be affected. And these are some conditions associated with it. So up till now, I've discussed the most common Mendelian forms of inheritance, but we must remember that there are many different and other types of inheritance which we call non-Mendelian inheritance, and these include multifactorial inheritance and triplet repeat, and genomic imprinting and uniparental disomy, and mitochondrial DNA mutation. So just to start with multifactorial inheritance, in fact, many of the conditions which afflict humans are multifactorial in nature. These include many of the neurodegenerative conditions, many of the old age or advanced age conditions like heart disease, cancer, and some examples that may be detected in prenatal diagnosis like pyloric stenosis, neural tube defects, cleft lip and palate. With these conditions, the ones we call multifactorial, there's a combination of genetic and environmental factors. And usually some genes are involved and some environmental factors are involved. And there is a threshold. So when there is enough accumulation of factors, genetic and environmental, a certain threshold is passed and the individual is affected. But if the individual has only some environmental or some genetic condition, then the condition is not manifest yet. Triplet repeat expansion is another non-Mendelian form of inheritance. This is caused by an expansion of a certain triplet. Say this is the gene for Fragile X syndrome, and there is an element which is repetitive, a CGG element in this case, repeated a certain number of times. So normal individuals will have up to 55 repeats. Affected individual will have a full mutation more than 200 repeats. That causes the syndrome to occur. And then there's a gray zone in between. And with this form of inheritance, the individual who inherits the number of repeats may have inherited more repeats than the carrier parent. So with subsequent generation, the number of repeat expands to the point where the disease becomes apparent, so there's a certain degree of anticipation. And some diseases that are inherited in this form may include Huntington's disease, Fragile X, which is on the X chromosome, myotonic dystrophy, and so on. And with each condition, there's a different triplet repeat that is expanded number of repeats as it expands through subsequent generation, so does the severity of the condition. Another concept which is different from what Mendel has envisioned is called genomic imprinting. With Mendel's theory, both copies of the gene from the father and the mother are expressed to the same extent. And that is true for most of the autosomal recessive conditions and the dominant conditions that we've described. But it appears that that is not true for all types of genes. Some genes are imprinted, that is to say, they are only expressed from one copy of the gene, from one allele, whereas the other allele from the other parent is repressed or imprinted or silenced. These are all synonyms. So an imprinting usually occurs by having one copy of the gene methylated. That means a methyl group is added on to the DNA structure and causing it to be more compact and non-expressed, where the other copy, which is non-methylated, is loose enough to be expressed. So only one copy is actually active. And it's important to recognize that there is a parent of origin difference. So some genes are expressed only from the paternal copy. Some genes are only expressed from the maternal copy. And the imprinting is erased. So the parent of origin is erased in the germline and then reestablished in the embryo. And then is maintained during mitotic division. So that's an important concept. This leads to the next issue, which is called uniparental disomy. This gets a little more complicated. Okay, so with uniparental disomy is a situation where both chromosomes are inherited from the same parent. We know that usually each copy of a chromosome is inherited from the other parent. But with uniparental disomy, due to situations that I will explain in a minute, both copies come from the same parent. Now, this is clinically important if that chromosome contains genes that are imprinted. And that it also increases risk of certain autosomal recessive condition. Now, I'll walk you through two examples of uniparental disomy. One is called isodisomy, where both the two copies of the homolog of that chromosomes are inherited from one parent. They're identical. And heterodisomy, two different homologs of the same chromosome also inherited from one parent. So I'll give you one example just to clarify. Let's take an example of an oocyte which has undergone non-disjunction. So let's assume this non-disjunction for chromosome 15. And this is the oocyte. Instead of having one chromosome 15 from the mother, it has two chromosome 15. They're all in red. This is a slightly different shade. Okay? And when it is fertilized with the paternal haploid sperm, we will get a trisomy 15 conceptus. Trisomy 15 usually don't survive, so that conceptus will result in a miscarriage, unless there is some sort of correction. So correction may occur by loss of one of the extra 15 chromosome. So let's assume that the chromosome that is lost by correction is one of the maternal extra chromosomes. So let's say this red maternal chromosome 15 is lost. We're left with two chromosomes, one paternal, one maternal, no problem. That is, if all the cells in the embryo have undergone that correction. If only some of the cells have undergone correction, we will have a mosaic. Some of the cells will have the trisomy, and some of the cells will be normal. But assuming all the cells undergo correction, that is, in 2/3 of the cases, that will be normal. 2/3, because 1/3 of the cases, this chromosome 15 will be lost, and in 1/3, this chromosome 15 will be lost. But let's see what happens if the paternal chromosome 15 is lost. In this case, we have uniparental disomy. That means both chromosome 15 have been inherited from the mother. Chromosome 15 contains many genes that are imprinted. And in this situation, The condition that will occur is Prader-Willi syndrome. Conversely, UPD may also occur in a situation that's called monosomy rescue. Let's just imagine an oocyte with a missing chromosome 15. So it has only 22 chromosomes, and chromosome 15 is missing. That sometimes occurs. And then when that is fertilized by a haploid sperm, we will have nullisomy 15. That means an embryo with a missing chromosome 15. Again, this is non-viable unless something occurs. And what occurs in this situation could be a duplication of that same chromosome 15. So we'll have two copies of chromosome 15, but this time occurring, both of them having been inherited from the father. And this is isodisomy, chromosome 15, uniparental disomy. In this situation, we get a condition called Angelman syndrome. And again, this also may be in a mosaic form where some of the cells may still have nullisomy 15, especially if evaluated early enough, say with preimplantation genetic screening. And we'll hear more about mosaicism and UPD in other talks. Lastly, a little bit about the mitochondria. The mitochondria are the intracellular particles. They are just like batteries. They produce the energy for the cell. And interestingly enough, the mitochondria have their own DNA. So each mitochondrion has several copies of a DNA molecule. It's a circular DNA molecule having only 16 kilobase long, and it encodes 13 polypeptides of oxidative phosphorylation, which is responsible for generating energy. But here's the important thing. The mitochondria are inherited only through the maternal lineage. And they're inherited only through the mitochondria that are present in the oocyte. The few mitochondria that are present in the sperm are lost. So all mitochondrial inheritance is exclusively maternal. And mutations in mitochondrial DNA may cause different conditions, usually affecting muscle and nerve. And here's in some of the condition. And also something that is important to understand, that not necessarily all the mitochondria in the individual are affected to the same degree. So, for instance, if this is the mother's oocyte, where some of the mitochondria are normal in green, and some of them carry mutation, this mother may have certain offspring. Some of them will have a high degree of mutant mitochondria, and therefore may cause a serious childhood onset disease. Some of the oocytes will have only some, say 50% mutant mitochondria, so that may cause a later onset disease. And some of the cells will inherit only very little, so this may be an unaffected individual. And these are some examples of mitochondrially inherited conditions like myopathies or optic neuropathies and so on. So with this, you have graduated Genetics 101. So if anyone has a question, here's the time to use this. Ariel. Yeah.
Cool, but I have no question.
Okay. Don't be shy. Okay, so if anyone wants a question, we send you one of these. You don't have to get up. You just have to speak directly into the mic. All right? Anyone? Okay, so I assume that everything is understood. So with this, we continue with the next talk.